you're using like lab experiments, but also animals and humans. And there, a lot of problems arise in a later stage during the verification and validation stage, right? So, however, the later the stage is, the more expensive it gets to solve a problem. So during the validation stage, it can be 1,000 times more expensive to make any design changes than during the concept stage. Now, with in-silico solutions, so using computational modeling and simulation, it's possible to find and resolve a lot of these problems early in the product development where costs are still low, but also to increase the evidence and confidence throughout the whole product development process. Therefore, the use of in-silico methods can give results more quickly and economically, and thereby de-risk the whole process and also bring the devices faster and more secure to the market. And these computed solutions can not only be used for development work, but also for regulatory evidence to prove safety and performance of the device. In the recent years, the so-called in-silico evidence has become not only a high priority, but is now established as a regulatory science in the US, Europe, and beyond. There have been some significant guidance documents that have been introduced that you can see here, notably contributed by the FDA, ASME, and MDIC, who are strongly supporting this approach. These documents then address critical aspects such as the reporting and credibility assessment of the computational modeling studies in the context of the medical device submissions. Now, how we address this at Retonomy is in two areas. The one aspect is to perform non-clinical assessments, so to simulate the physical device to generate this evidence for safety and effectiveness. This is to reduce in vitro testing, but also to reduce and support in vivo animal testing, maybe also to prove substantial equivalency with non-clinical data. The other area is then to augment clinical assessment, so to evaluate device performance using a virtual patient cohort that is then with simulated patients. This is then to have trial planning and close this evidence gap of diversity, but also test on a diverse patient cohort, like really detached, underrepresented patient cohorts. And by that, also tend to mitigate the risk of failure modes, as I said, then to reduce the risk to fail in a later stage. We're doing this by starting with data, so real patient data. Here we've collected a diverse database with our clinical partners regarding pathology, ethnicity, age groups, and also we're including animals in this. With that, we can then create three-dimensional representatives of the population, so this so-called virtual population. This can then be combined with virtual benchtop testing and also advanced analytical simulations. So as a first step, we can provide here statistical population analysis to better understand the patient cohort. So to create rationale for worst-case assessment and verify inclusion-exclusion criteria, because you're not developing your device just for the average patient, but also then you want to identify these critical areas. And as said, this can then be combined with this anatomical simulations that will show then later on how this works in detail. And finally, you create digital evidence can be used then for the regulatory submission, where we are compliant to the guidance document that I've just shown you. So how does this look in action? So here you can then, as a first step, select the patient cohort. So create your virtual patient based on your target group that you want to address. You can then upload your own device, include this then into the patient. So for example, here first position it at a certain location, and then in an easy environment, you can select then also the conditions you want to have for the simulation. So next step, you can then access also the statistical analysis. So as said, identify maybe the worst case assessments. Like here, then a multitude of patients. You can see, you can cluster it in male, female patient, but also in different pathologies, diseases, different measurements that you perform there or that automatically are performed, to then have this density curves also to access like the statistical of the population. Then this can be combined then with non-clinical assessment, like here, for example, to assess the hemodynamics of a heart valve, and also fatigue analysis of a stent, combine then also the two different isonorms, or also the cannula pressure drop analysis to assess hemolysis or blood clotting. And then as a next step, this can then be performed and combined with the anatomical data, like here with an LAO occluder device, but also the catheter application. This is an example I want to show next in more detail. And then finally, also what is possible is to use electrophysiology to even include the beating heart in the assessment there under different conditions. So let's go to a use case. So to an example of how this can be used, in this case, transcatheter devices. First, what are transcatheter applications? So there's a multitude of applications. So via transcatheter, you can have a valve replacement, as you can see here with the Medtronic Evolute. You can also have transcatheter repair system. There's the tricuspid valve, like from Abbott, or even a blood pump that is minimal invasively implanted, like the illuminate impeller system. There are various implantation access routes, like the transfemoral access route, transaortic, transcarotid. Where here the transfemoral access route is the most favored one because it's the least invasive one when implanting the device. Now, when developing such devices, there's a lot of challenges, especially coming from the anatomical variability. You want to then have the correct design, the material selection, but also the implantation insertion protocol that is there to then avoid adverse events, but also then access even smaller vessels that are more tricky. And this is very difficult to test when you use experiments because these are usually very simplified in a tube. Animals, they're very different from anatomy. So a lot of issues arise in a later stage. Issues can be like the risk of bleeding when you then have the anatomical variability in there, material failure with the bending, or other aspects like vessel or organ injury in there. So our process in this case is then to have a database of patients that are coming also from the transfemoral access route, where we can reconstruct then the anatomy based on this, do statistical analysis in our system, where we then access here, for example, tortuosity, so that patients with low tortuosity, patients with high tortuosity, we can then have also the percentile of these patients included there. And these specific patients then from the database can then later on be accessed also for the simulation. This is then the next step where we can have the catheter implantation as one example in the lower limb, but also then in the aortic arch then towards the ventricle and to the deployment area. We can not just look at the transfemoral access route, but also other access routes like the inferior venous system. You can see to the left, in the middle, then the subclavian and jugular access vein, as you have also seen at the beginning, or even more complex access routes like the transeptal via the fossa ovalis that is then implanted then through the wall between the atria. When we do the statistical analysis, there's a multitude that we can access. Here, it makes sense to separate the vessel into smaller regions to make this also more comparable between all different anatomies. When we run this through 100, 200, 300 patients, to the right, you can then see like in these different areas, then also like the diameter, lumen diameter for the average in green, the patient, but also then the smallest and largest. Between the smallest and largest, this is where you then also have your target area, or what can also lead to critical zones then for the implantation. We can also look at calcifications there, like the volume of calcifications that is critical when implanting such a device, but also then differentiate between circumferential calcification or longitudinal calcification, as this is also critical when implanting these devices, because this then also changes the stiffness of your vessel wall when you have the vessel straightening with your device then when it is implanted. And this vessel straightening, this is what we can then simulate. So what we provide there is a tailor-made simulation environment that is customized to your specific needs in there. So we are then considering all the device components, like in this case, like the guide wire, the sheath, the catheter that's implanted with the specific material properties from the device itself. Then also the catheterization process. So we are following here the clinical practice. I will show this also there with the different body conditions that is included. We also have advanced arterial material modeling of the vessel wall and also the calcification. And we also take into account the surrounding tissue and organs when we have the vessel tissue or the device tissue interaction, to also then have the interaction with the surrounding organs and also then the bones to then also really simulate like how the vessel is really straightened there. Regarding the tissue material model, we then also have complex models included there, where we consider also the different layers that is included in this, but also the inhomogeneous soft tissue, like a hyperelastic material model, also then locally varying fiber arrangements, so also an anisotropic material that is considered here. So also then the complexity of the tissue can be included in the simulation environment. Also the complexity of the implantation process itself. So the catheter insertion, also different angles. We are following here the clinical guidance, like the Seldinger technique in this case, where we introduce the guide wire followed by the sheath and catheter, and then also the cutout portion in the skin that is also considered here in there. And the user can then identify the specific location then for the implantation. Now, finally, everything gets together. So the user can then, when you're interested in different design sizes, for example, of your device, like a 17, 19 French cannula or catheter, for example, or a different stiffness in this case, you can then combine this with the anatomical variability of all this population, like in this case, the iliac artery, with then the different dimension, lumen, tortuosity, so simulate over these, and then really have like a design of experiment over all these populations and the designs. So you have the design parameters, you have the anatomical variability, and then the outcome that you're looking in. So your question of interest that you're interested in, maybe this can be relating to bleeding in this case or other aspects, and then have either a design optimization there, or you're interested in a worst case assessment for inclusion exclusion criteria, an extension of your IFU or whatever you're interested in, and use this then as justification for the regulatory assessment or for extending to your clinical trial. Another example I want to show is then the advancement to the heart valves. So when you have the catheter, you're then deploying this into the different valves. We have here also gathered a big database over all these heart valves, so mitral valve regurgitation patients, tricuspid valve. You can see here also over the whole cardiac cycle with in-systolic and diastolic, but also the cardiac cycle included. Again, to the right, the statistical assessment, in this case, then the annulus plane and other areas that is of interest. Again, you see then the smallest, largest, and also average diameter then of the annulus plane, also considered with the end-systolic and diastolic phases there. As next, we can then also access like benchtop testing. So this is then to evaluate, for example, the hemodynamics. In this case, here then for hemolysis or blood clotting, this is typically assessed using then the ISO 5840 benchtop testing, but we can perform this as a so-called fluid structure interaction simulation to really simulate the leaflet dynamics and the blood flow in there. But it's also part of the ISO 5840, so to use the simulation environment. It was validated against particle image velocimetry, very good accuracy in the lab that we are working with. After the validation and then following the verification validation of the simulation, we can use this then for accessing then the device itself, but also then not just under this perfect ground condition that you typically assess in an experiment, but then really take into account like all the statistical analysis from the population and using this for the simulation. As next, we can also simulate it specifically into the patient environment. So we are following then here also the clinical process, like crimping then the stent. So here, in this case, we're only looking at the stent, but also the leaflets can be included. Then as next, we can look into the release into the device or into the device into the specific patient in there, like then here from the different views. And then even take into account the contraction, expansion of the environment, where you can see to the right, then also with the different conditions of the heart during end-systolic and diastolic, and use this then also as a condition for further analysis of fatigue. And here I want to show one example of regulatory submission from one of our users that we are allowed to show. This was for a compassionate use case submission in Germany for the first in human submission. This was a very severe patient, and there was only 3 weeks time window for the submission for this specific patient. And we performed then in silico all the fatigue for this, had some in vivo data of a predicate device. We're then following the whole FDA guidance on the reporting, but also the verification and validation was submitted to the BFARM and accepted, and then also accepted for the implantation into this specific patient. Now, only a short summary, but I think we are also at the end of time. So potential impact, I think, was clear. This can be used for device development and clinical trials in all different stages of product development and become a complete workflow in the device development, but also then in the preclinical and clinical phase then of your device that can be used for the submission to the bodies. Thank you very much. You can see here are my contact details. So if you're interested to learn more, feel free to contact me or our website, but also looking forward if there's any question and when we have time for any discussion. Thank you.
Thank you, Simon. Let's go. I have a question. I think in your bio, which I will share in the chat box, that you said 40% of the devices can use in silico simulation. But what kind of devices can actually use your software to validate, like what you just shown, and what kind of devices we cannot?
Yeah, so we are specifically focusing on cardiovascular devices. So
Okay.
this is everything that is relating to the heart. So we are looking at stents, but also like assist devices. Here, I'm currently in Krakow at a conference for ECMO, so
Okay,
oxygenators, cannula systems, where this can also be used. So we are looking very much into everything that is implantable and inside the body. It is also possible to use for other devices like class 1, but there maybe the need is not that much. So for everything relating to class 2, class 3 devices, this is something that is very relevant to use such in silico simulations.
And then in terms of the timeline contraction, using in silico simulation for a regulatory process, what is you give us an idea of what is the contraction like? Okay, now this is a 3 weeks, so you got it through FDA. What would it be if we don't have in silico?
Yeah, so in this specific case, it would have not been possible to implant in this patient because they were not able to generate any other data. Like in vitro, it would have been possible in this time frame to generate data that is